as we go, but I do want to get it started because we have a lot to cover today. I'm Jolene Simon with Koochiching Soil and Water Conservation District and co-hosting with Lindsay, I think I said that right, from MPCA, and Kimlin Koviak from Itasca SWCD. So today we have quite a bit to cover. It's our first meeting for Big Fork River Watershed in quite a while. I want to thank you for coming to this virtual meeting. It's a different way to do things. We do have quite a few presentations. We have 5 people speaking today. Lindsay will be speaking. We'll have Megan, Jesse Anderson, Megan House out of MPCA, Jesse Anderson from MPCA, and Rachel Lindgren on Springs from Minnesota DNR, as well as Kimlin Koviak. All right. So I'm going to go ahead and get started with our housekeeping. And for today, because we have a lot of people on and not a lot of time, if you're having technical issues, feel free to connect with Lindsay via chat. She will help you get situated with that, or I will try as well, but she is on the line running the show here. So this session, just so you are aware, will be recorded and posted on our website after the fact, either by the end of the day or probably more about tomorrow. Everybody was muted on entry. We'd like you to please stay muted while presentations are happening, and you can unmute yourself to speak though if you would like. Having your camera off during this is much better for bandwidth for all. Some people are in lower bandwidth areas. Please hold your questions. You can type them in the chat at any time, absolutely. But for now, because we want to keep rolling, we'll hold questions until the end, unless we find we're rolling really fast. So again, type them in the chat at any time and we'll get to them as quickly as we possibly can. So without further ado, I'd like to pass it over to Lindsay from MPCA with an update on Cycle 2.
Okay, thank you, Jolene. So I'm Lindsay Cremery. I'm a watershed project manager with the Minnesota Pollution Control Agency out of the Duluth office. And I am the project manager for the Big Fork River Watershed. And so today I just wanted to introduce to you the Cycle 2 Watershed Restoration and Protection Strategy process, and then give a little brief overview of Cycle 1, which happened 10 years ago, and then also go into Cycle 2 projects and updates. So you can advance the slide, Jolene. Thank you. Okay, so just a little brief recap of Cycle 1. The last monitoring and assessment happened in 2010. So the sampling and the monitoring happened in 2010 and 2011, and then the assessment process happened in 2012. And so we do our RAPS process, which RAPS is the Watershed Restoration and Protection Strategy process. We do this every 10 years. And so Cycle 2, we're just coming up on Cycle 2, and this is kind of the kickoff to that. So, but for Cycle 1 overall, the Big Fork has good water quality. The assessment results for the watershed indicate that the condition of the lakes and streams are good to very good, even though there were a few impairments found. Can go to the next slide. Okay, so the locations monitored by MPCA, citizen volunteers, and local partners, you can see in the purple dots on this map. I know it's pretty hard to see, but basically the gist of it is just to kind of notice how many purple dots there are. We had a lot of sites. In Cycle 1, it was our goal was to evaluate the whole watershed and the condition of the whole thing and kind of get that baseline monitoring. So we really had a lot of sites. And so we examined the water quality and then identified stressors, and these informed the Watershed Restoration and Protection Strategies report. And then we use this to track trends also. So you can go to the next slide. All right, so just to give a brief overview of the impaired waters, the most widespread impairment found in both lakes and rivers is due to high mercury levels. limiting the aquatic consumption of fish. So 12 lakes were impaired for the aquatic consumption of fish, and then the big parts of the Big Fork River. The remaining impairments throughout the watershed consisted of low dissolved oxygen with impairments in 3 streams, fish and macroinvertebrates with impairments in 9 streams, and then the nutrient impairments in 6 lakes. And many of the aquatic life impairments were found to be the result of natural conditions within the Big Fork River watershed. You can go to the next slide, Julie. Thanks. So if you're interested in seeing more of the details on, which lakes passed the assessment and which ones were impaired, or to dive in any deeper, you can head to our website for the MPCA and then go to the Big Fork River page, and I'll be sending everybody out an email with this link afterwards, so you don't have to worry about trying to write it down. But here you can find all of our reports. We have the monitoring and assessment reports, which really goes really deep into the water quality part of things. And then we have some studies on Island Lake and then Jessie Lake. And then we also have the Big Fork Watershed RAPS report for cycle 1. And then this will all be updated as we progress through cycle 2. So we can go to the next slide. Okay, so just to give you some overview of how cycle 2 is going so far, we started the intensive watershed monitoring in this summer. So it started in May 2020. But due to COVID-19 restrictions, some of the work was not able to proceed as planned this summer. But so work that required boats or close personal contact was halted for MPCA staff. So this included the fish and invertebrate sampling and also the water chemistry sampling for lakes. And this was for MPCA staff. And then local partners were able to continue their monitoring contracts according to their organization policies. So we did get out and collect some data, and I will go into that in a little bit. But as far as next summer goes, depending on social distancing restrictions, the hope is to get all the rest of the lakes caught up, and then we will need to reevaluate and see if we can complete the fish sampling. But the hope is that we will be able to do that this next summer. So yeah, Jolene, you can go to the next slide. Okay, so how do these COVID-19 restrictions affect the Big Fork? Stream sampling was conducted in 2020. And so the monitoring was completed by Kuchiching SWCD. And then the lake monitoring was completed by Itasca SWCD and Leech Lake Band of Ojibwe. So there was 13 lakes completed and then 8 stream sites monitored. And then the chemistry in the lakes and then the fish and invertebrate sampling that were not collected in 2020 are slated for 2021. And then the watershed assessments are likely to be delayed by 1 year. So instead of 2022, they'll be done in 2023. We can go to the next one. And this is just a real brief overview. There's a map of the chemistry sites that were sampled this year. If you can see the sites in green were sampled. So there's a couple along the Big Fork there, one on the Bear River, one on the Sturgeon River, and then one on Caldwell Brook. And then the red sites that you see, those are our biomonitoring sites. And those are, we hope to get to those next summer. And we can go the next slide. And this is the rest of the Big Fork, the southern half. So this shows one more stream site in green, Bowstring River. So that was sampled this summer. And then the rest of the sites are lake sites. You can see they are in the dark blue. So those lakes are the ones that have been selected for this year. And the ones that were sampled already this year are in yellow. So you can see the lake names over on the left too. So Busty's, Muir, Bellow, Maple, Long, Trestle Lake, Rush Island, Bowstring, Little Sand, Round, Dunbar, and Moose. So those ones were done this summer, and the rest that you see there in the dark blue will be done next summer. And we can go to the next one. Okay, and so some additional Big Fork Cycle 2 work we have planned it is to do a Big Fork River assessment, so some stream bank assessment, and then a springs and headwaters stream monitoring project. And we want to look at water bodies that are close to impairment so that we can try and put some protection strategies in there to make sure they don't tip over into impairment status. And then potentially looking further into Island Lake and investigating the phosphorus situation there. So we can go to the next slide. And this is my last slide. It's just a project timeline. It just kind of shows we're starting in 2020. We're starting with the monitoring, and some of the other projects are already starting, like the Big Fork River Stream Bank Assessment and the Spring Survey are already started. But if you look, we have some of the reports going out until probably trying to complete these all up in 2024. So that's the timeline. And then we can go to the next slide. And that's all I have for you guys today. So thank you.
Can I make a comment, please, Dick Locker?
Yes, go ahead, Dick.
I'm wondering why nobody mentions the fact that we've been doing chemical monitoring in the river with the 2 high schools for 25 years now.
Oh yes, we have a whole part of our presentation on that actually today. So we definitely did include that data in the cycle 1 assessment. And so your data was probably one of those purple dots that we showed. And
well,
so
I tried to give you the data, and I have to change the whole format of my collecting, and I'm not willing to do that. I don't have the time to change the format. So you probably aren't even collecting my data anymore, although I have it.
Okay. Yeah, I mean, I can certainly get in touch with you after this and we can chat about that. That sounds good.
Okay. Incidentally, my data agrees with the PCA data. The river's in good shape.
Mm-hmm. All right.
Mine's cheaper than yours.
Lindsay, this is Lester Kaczynski. Can you talk about the phosphorus issue in Island Lake? Thank you.
Sure. So maybe I don't know, Jolene, if we want to take the questions at the end, or if I should touch on that real quick now.
I think if we could hold that until after, that might be best. Yeah. Is that,
and then if we run out of yep, that sounds good. If we run out of time too, I can personally follow up with you, Lester. That sounds like a good plan.
Okay, thank you. Thank you. All right. Well, thanks so much. I'd like to introduce then Megan Holthouse, and she will be presenting on the intensive watershed monitoring. And this portion is PCA driven monitoring. So Megan, when you're ready, take the word.
Hi, yeah, can you hear me?
Yes, you're good.
Perfect. Hi, everybody. So again, my name is Megan Holthouse, and I work for the Minnesota Pollution Control Agency doing water quality monitoring. And I am actually the lead water chemist for the Big Fork River Watershed. And yeah, so I'll be talking to you about the monitoring we're doing here, the parameters that we collect, and what we do with that. Next slide. All right, so first I'm just kind of going to talk about really briefly the whole watershed approach. That is involved with the monitoring. So the watershed approach is a 10-year process that creates a common framework for our water quality work and is an efficient, cost-effective way to address impairments. So we're able to determine the overall health of a watershed's water resources. It also helps us identify impaired waters and those that need protection to prevent impairments. The very first step of this process involves collecting data intensively for 2 years. So in 2020, we have started doing that, cycle 2 as we call it, after we've already went through our first iteration. That we reach the end of the 10 years, we're on to starting it again. But because of some setbacks, so normally it would have been 2020 to 2021, we would do 2 years of monitoring, but it's quite possible it could go into 2022 just because of COVID. And then all water bodies with available data in this watershed will be reviewed, assessed, and included in a watershed assessment report, so long as that data has been submitted to the MPCA. And then at the completion of assessments, step 2, then that is when we start that stressor identification. So we'll study the data and identify conditions stressing water quality, as well as those fostering healthy water. So that'll be other people in the agency who handle that. And then step 3 is the development of that watershed restoration and protection strategy, or RAPS, and local water, and then also local water plans to restore and protect water bodies. Then finally, with step 4, the project manager as well as locals will implement these projects and activities to restore and protect our water. And then we just repeat this after 10 years is done. So yeah, Big Fork has already been through the first iteration and we just started it. Next slide. So I'll talk about the biological monitoring that we do. So it's conducted by both the MPCA in streams and DNR in the lakes. So it's a one-time fish, invertebrate, and chemistry sample for the streams, and they also do a habitat assessment. And then fish and plants are sampled in the lakes, and this will provide a direct assessment for the health of the aquatic community. And this data, especially in the second round, cycle 2, is important for potentially delisting any waters. So any of those impairments, if we hope to remove them, if we see any progress, this could be great to possibly get rid of some impairments. We can also do problem investigations if, maybe there have been some fish kills or something. Also address stressor identification needs, or there's also permitting needs like effluent limits at the agency. Next slide. And then these are just all of the stations that we collect biology. So it's both fish and invertebrates. And then, so 45 sites were selected this year, 4 of which were in the Leech Lake Reservation, and 3 of which were in the Boy Fort Reservation. So all of those red dots there. Next slide. And then for stream chemistry, most of the monitoring is actually completed locally via contracts with the MPCA. So we have these surface water assessment grants, or SWAG, we call them. As you heard earlier when Lindsay talked, a lot of people have been doing monitoring in the watershed. I don't even think we had to do the MPCA. I don't think we had any sites because everybody picked them up, which is great. But whatever they don't pick up, then we'll take. And then this is 2 years of sampling, and that focuses on nutrients. So that'll be total phosphorus, chlorophyll A, and we have a few other parameters. And then sediment, bacteria, so that's E. coli. And then we have these sondes we use that are field equipment. They collect dissolved oxygen, pH, and temperature measurements. And this data can also be used to potentially delist waters, also fill gaps identified by modeling, track changes for any waters that are maybe close to being impaired or maybe close to being delisted, and then also informing permits and looking into any potential problems in the watershed. Next slide. And then these are the sites. So there are 9 sites that are being monitored. And the green dots there are the chemistry stations that the MPCA or SWAG are doing. And then they might look purple or pink depending on your screen settings. But that triangle there is what we call pollutant load monitoring. And that those are sites on the streams that the MPCA is sampling long-term data on related to storm events. So that's even more data that they collect on a long-term basis. Next slide. And then for lake monitoring, water chemistry is also completed by MPCA staff and locally via contracts. And this could be 1 to 2 years of sampling. That just might depend on how much data we already have on the lake. If there's no data at all, we might do 2 years. Depending on if we think there could be a new impairment, we might do 2 years as well. And then this data focuses on nutrients, algae, and clarity. And then again, just a reminder that DNR samples the fish and plants on a subset of lakes. And this can also be used to delist waters, look at changes, and look into problem investigations. Next slide. And then, so on the other, I have another page, the next page will show a list of the lakes, because there's 32 lakes, so I'm not going to list them all here, but basically all lakes in the southern portion of the watershed there were chosen just because they're like a little more recreationally significant. They're bigger, more people likely to be on them. They're easily like accessible to. So 32 lakes, 6 of which fall within Leech Lake, and then 1 falls within the Boy Fort Reservation. Next slide. And then I won't really read them all off here, but as you can see, there are all the lakes. The stars indicate lakes that fall on the reservations. As you heard, Bowstring, Rush Island, Coon, Sandwick, a lot of those lakes are getting sampled. Next slide. And then this is a map the DNR provided me with of the lakes that they did biological monitoring on. So 14 lakes were planned and 10 were surveyed between 2016 and 2018. And then 4 were surveyed in 2019. So just to call out a few, Coon Sandwick was done, Pickerel, Mirror, Deer Lake, Sand Lake, Rush Island, Jessie, Little Jessie. All right, next slide. All right, so all of that data, I listed off all of the things we collect. The next step of that is once we have all of our data collected, then we have to take a look at whether it does or does not support aquatic life use or aquatic recreation use. And so when I say supports aquatic life use, because I realize maybe not everybody is aware, we're wondering, does it support a healthy fish or bug community? Can they survive there? And then in regards to aquatic recreation, is it safe to swim in there, to fish in there, essentially just touch the water? So out of those parameters I listed for aquatic life use, the fish and macroinvertebrates biology sampling, that's part of aquatic life use. So it's dissolved oxygen, pH, total suspended solids, river eutrophication, so that's that phosphorus, chlorophyll A, also biochemical oxygen demand, and then we also have unionized ammonia and chloride. And then for streams, for aquatic recreation use, that's that E. coli. And then aquatic recreation use is a little different for lakes. That's just that total phosphorus, chlorophyll A, and water clarity measurement through the Secchi disc. And then aquatic life use on lakes is fish, IBI, and chloride. Next slide, yeah. So then just to break down a little bit like the assessment process, just so you understand kind of how we get where we do. So we start data collection and processing. So we'll have a call for data. It's usually the fall of that second year of monitoring, but again, because of COVID, it's possible we won't be doing that next year. We might not call for data until 2022. And then once we have that data and everyone submitted it and reviewed it, it'll be stored in our database called Equus. And then that data is then imported into a database that we use that summarizes our data and lets us do assessments on it. And then we do the desktop review next. So we'll do an individual parameter by parameter review. So biological data is reviewed by the invertebrate and fish biologists, and then the water chemistry data, I look at that, reviewed by water chemistry staff. And then we review these independently at first, and then the end result is a determination whether each parameter meets or exceeds the applicable standard. And then the very next step after that is once we've done our initial desktop review, we take it to an internal team. We call it a watershed assessment team, or WAT. And so that's a meeting of internal PCA staff. So biologists, chemists, stressor ID, project managers, the pollutant load staff, anybody who kind of played a role internally in sampling in this watershed is there. and we try to determine an agreement on use determination. So, yep, I agree that is an impairment for aquatic life for dissolved oxygen on the stream. Yeah, and then we do aquatic recreation, and then let's see, preliminary assessment decisions are made there. And then we take that one step further and then have a professional judgment group with the public. So usually it's more watershed district folks, soil water conservation district folks, anybody else who is a scientific community that goes to these. Anybody is welcome to come. It just does get to be a little overbearing because it's a lot of data and talking through each stream, so it can be a little boring. But so we meet there to go over our initial assessment decisions and see and get opinions. Does anybody disagree with this? Does anybody have any input that maybe, we didn't know this certain thing was happening on this stream and it would be good to know? Or maybe there's new data that somebody didn't submit that we could look at instead before we go and put an impairment on something. And then once we have the PJG done, then we have a public comment period that we send all of the impairments. So the public comment period is for the impaired waters list. So anything that we found to be an impairment will end up going on there. And that's every 2 years. And that's with the EPA. So anybody, again, that's like our very final last chance for anybody to make a comment on whether or not they agree with impairments or ask for information or provide information. Next slide. And then just this is my last slide, kind of to draw one more connection of maybe last year. So last year we completed the full iteration of cycle 1 in all 80 watersheds in Minnesota. And there were big headlines about the state found 56% of Minnesota's lakes and streams are impaired. So basically the work I just talked about here is a big part of what goes into that. I believe only 30% were actually based on the parameters I just spoke about, as we do have mercury, drinking water standards that also go into impairments. But yeah, so that's just kind of how we get there. And that's all I have. And if you have any questions, you can definitely call me or email me as well.
Thanks so much, Megan. Excellent. I welcome anyone to chat your questions right now if you have them. We're trying to save some time for questions at the end for any of the speakers, but we will have the opportunity to respond later as well. So without further ado, Jesse Anderson of MPCA.
Can everybody hear me okay?
Yes, you're good.
Okay. Hi, folks. My name is Jesse Anderson. I'm a research scientist with the MPCA in the same group as Megan out of our Duluth office. I've been working in the area for quite a while. Nice to see some new and old friends on today's meeting. I'm going to spend a few minutes and talk to you folks about blue-green algae, sort of public outreach as it relates to lakes and protecting public health from when we have too much algae in lakes and what we can do about that. So next slide, please. I'm going to start out with just a few summary talking points about blue-green algae. They are a native algae. Some people think they're an invasive species like zebra mussel. That is not the case. Blue-green algae are present in most waters in the world, and they are present naturally in all Minnesota lakes. They are a group of algae that tend to grow best in hot, sunny, calm conditions, sort of ideal conditions for us to swim in lakes. And when they grow to excess, they can grow in large mats that we determine as harmful algal blooms, or HABs. What's very important about these groups, this group of algae, they're very historic organisms. They're some of the first life forms on Earth. They've been around for billions of years, and they produce toxins during some portions of their life cycle to give them a competitive advantage over other types of algae. And one of the big hot topics in the lake research world right now is, and there's a lot of smart people all over the world trying to figure this out, are what are the conditions environmentally, whether it's temperature, um, chemistry conditions, or other things that cause them to produce these toxins. And we don't know that at this point, but these organisms produce toxins that are that can be toxic to the internal organs of livestock, pets, and humans. One of the first confirmed incidents of poisoning is from Minnesota in the 1880s, where a bunch of cows were killed after drinking tainted water from a farm pond. So people have known for a long time that these types of algae can be detrimental to us and our pets and our livestock. There is no way to tell by looking at an algal bloom if it's producing toxins that can make us or our pets sick. I'll get into that in a little bit. But again, these excessive algal blooms or harmful algal blooms are most common in lakes that are higher in nutrients, but they can occur anywhere, even some of the most high quality lakes in this watershed and in this area. I've seen algal blooms in remote parts of the Boundary Waters Canoe area. They've been documented in the really clear deep lakes in Isle Royale, where there's no human impact whatsoever. People think it may be related to climate change, but these blooms, they come on during the environmental conditions that they grow best in, hot, sunny, and calm weather. They do naturally dissipate with wind, waves, and rain. But a very important point is that these toxins can still be present in the water, even if you don't see the visible greenness to the lake. When the algae die, the cells break apart and the toxins are released into the water, and they are not colored green. They can still be in there. So another question we often get, well, can I just boil the heck out of the water and still drink it? The answer is no. Um, Basically, we're here to just advise folks to be cautious about these conditions, and I'll get into that in a little bit. So next slide, please. So this is a typical picture of what an algal bloom looks like. This is a lake in the Big Fork Water, Bear Lake in the headwaters of the Bear River. In our watershed, this is a most productive lakes in the watershed. This is a bloom of microcystin. This is the type of conditions that we're trying to get people to be aware of. Obviously you can see the nature of that bloom there. Sort of a green growth of algae on the surface from, again, early August, kind of the dog days of summer, when we tend to see these most typically. However, we have incidences even into the late fall on some lakes. Next slide, please. So the one thing that I ask you folks to remember is to take a real conservative practice. When you see conditions like the last slide, it's best to stay out of the water and keep your pets out of the water. When in doubt, stay out. And we recognize this can be a challenge because dogs love recreating in lakes. They love smelly, stinky water, and it can be tough to keep your pets out. But I hope to convince you that it's important for you to do that if you see an algal bloom. You do not have to be an expert in lake science or environmentalism to see these blooms like the past picture. Again, a very obvious green paint-like appearance on the water surface. You often get a strong musty odor. The lake doesn't smell good. It doesn't look good. It may be accompanied by a fish kill, again, by these toxins that these algae produce. And again, they're most common in mid to late summer. These algae love it when the water temperatures are at their warmest, which be uh, end of July, early August in your part of the world. They love it when it's hot all over the world. So, but if you or your pets come in contact with the harmful algal blooms, wash them off completely. Please don't allow your dog to get out of the water and start preening their fur. We have several incidences, unfortunately, where dogs have passed away. Um, we've traced it back to algal blooms, and confirmed by veterinarians and epidemiologists. We think what happens, these dogs are preening their fur, the algae is stuck on their fur, and they're getting a concentrated dose of algae in their bodies. And we have, again, unfortunately, several cases of dogs dying, sometimes within hours of swimming in a surface water body that has a severe algal bloom in it. They often vomit immediately, and again, in rare cases, can go into convulsions. These toxins, the most common one affects the liver. So if you see this on your dog, please hose them off the best you can and get them to the veterinarian immediately. And if you have some symptoms, for us, we're probably not going to be ingesting a lot of this water. It's most often going to be, sort of a dermatitis, like a skin rash, irritated eyes, or sore throat. Just stay out of the water. But if you're experiencing symptoms, get medical attention yourself. Again, we want to protect our lake users, the public out there. I'm trying not to be alarmist out here, but we have had several dogs pass away over the last decades, and a few incidents of human illness that have been traced to recreating in these conditions. So tons of information about this on our website. When this is posted online, I encourage you to go to that website and read more about how to protect yourself. But again, the main message is when in doubt, stay out. Next slide, please. So we have a triage or an incident respond process we've sort of been learning as an organization over the last 15 or 20 years as these incidents of pet illness, pet death, dog death, and rare cases of human illness have come our way. So we want to know if you see conditions in your favorite lake. If you see an algal bloom, please report it to us. Send an email to that email address listed on the screen, or call that water quality monitoring line phone number listed there. It's answered during business hours, Monday through Friday. If you call on the weekend, that's when most folks are at their cabin, please leave a message. We do to track. If you're more comfortable and familiar with your local county soil and water district office, you can contact them as well, and they can forward on the citizen inquiry to us. But we do want to know if you see an algal bloom on your lake, where it was, and the dates. Unfortunately, we are not able to respond to every reported algal bloom in the state. We just don't have the resources to do that. Us and our partners have done a great job, as Megan talked about, monitoring lakes across the state, but we've unfortunately had to develop a triage process where our staff, the MPCA, can only follow up with toxin sampling, taking a sample of the algal bloom, sending it to a laboratory, analyzing for these toxins, only if there's a suspected human, a dog illness, or a dog death. So that's why it's important for you, if that's happening to you or your pet, to please let us know. And we try to get out there as soon as we can. If you just are concerned about an algal bloom and want to report conditions like that picture I showed a few slides back, we do not unfortunately do not have the resources to drive out to your lake and confirm the presence of an algal bloom. There are things that you can do, and I'll get into that. We have this simple jar test, which is on our website, where anybody can do that. And there are other courses of action. There are some commercial labs out there that provide test kits to get a range of the most common algal toxins, but they can be tough to deal with and give, in my opinion, marginal results sometimes. And it's not a substitute for laboratory tests. There are labs in the state that can run these algal toxins, including RMB lab in Grand Rapids. But again, the samples have to be collected timely, and we can assist you with that if you like. Next slide, please. I'm going to wrap up shortly. So if you want to know if you have those conditions in your lake, the lake's green, looks bad, smells bad. Well, it can produce toxins that potentially make you and your pets sick. There's a simple thing you can do called a jar test. Take an ordinary mason jar that you have in your home or cabin or any glass jar. Take a sample of the algal bloom near shore or wherever it is in the lake. Put it in your fridge overnight and sit in the fridge for at least 24 hours. After a day, open up your fridge and take it out and look at it. And at the bottom of that slide there, if the algae settle to the bottom, it's likely not these blue-green algae that, again, tend to float to the top because they're able to do that and produce toxins. If the algae sinks to the bottom, it's another type of algae that likely does not produce toxins. If most of the algae float to the top, like that photo on the right, it's likely that you do have blue-green algae species. And again, stay out of the water until the algal bloom naturally breaks down. I think I have one more slide. Next slide, please. Just want to just share with you folks these incidents since we started really tracking this 15 years or so ago in response to some dog deaths that were reported to us in some very productive lakes in southern Minnesota. At that time, we started tracking this. We were very concerned about it. We formed committees with the health department and veterinarians to try to get the word out. The last 5 years or so, we've really tried to ramp up public awareness. That's one of the reasons why I'm here today. And this is just a breakdown by year of how many algal blooms are reported to us, how many dog illnesses, dog deaths, and human illnesses that we have recorded in our system. This is not an exhaustive list. This is just conditions that have been reported to us. Now, one of the things that we're trying to tease out, and so are a lot of other lake folks across the country, is are algal blooms becoming more common because of an increase in public awareness? In other words, they've been there all the time, but just because folks are more aware of them, they're reporting them more often, or are they actually becoming more common because of things like climate change and warmer surface temperatures, warmer, shorter ice durations that promote growth of these types of algae? So when in doubt, stay out. If you see something, take a picture, send us an email. And if your pets get sick, please contact us. We want to help you. So with that, I think I'll close.
Jesse, thank you so much. That was great information. I see Lindsay has been posting that email in the chat for everyone if you do need it, and we'll have all of this available after as well. Next up is Kimling Kowiak presenting for Shannon Martin, who is not available, on Citizen Lake Monitoring Program.
Thanks, Jolene. Yep, I'm going to basically just do Shannon's presentation as written from her perspective within the MPCA. But I, as an SWCD person, have worked quite a bit with these people in the MPCA who work with the Citizen Lake and stream monitoring programs to get lake associations, connected with the data portal people to volunteer their own lakes and streams. Next slide, Julie. So citizen science is simply described by National Geographic as the practice of public participation and collaboration in scientific research to increase the scientific knowledge. Next slide. The MPCA has been deeply involved in citizen science for decades, having administered 2 citizen water monitoring programs, one going back 45 years. And today we have over 1,300 volunteers that track water quality on lakes and streams across the state through these programs. Next slide. We offer 3 citizen science opportunities with the MPCA, a citizen lake monitoring program, a citizen stream monitoring program, and a smaller scale lake ice reporter program. Next slide. These programs generally focus on basic water quality using a Secchi disc for lakes and a Secchi tube for streams. Volunteers are asked to monitor at least twice a month from May through September, and even go out weekly if it's possible. Volunteers also can rate algae levels for lakes and sediment levels for streams, record watercolor and stream levels, and just some basic observations while they're at the site. But an important component of this program is that volunteers return to the same site every single time to ensure the consistency of the data they're gathering. Next slide. The Citizen Lake Monitoring Program also does offer a BWCA monitoring component, which is different from the standard lake program in 2 ways. And first, a volunteer doesn't have to participate for the entire season, but just monitors a lake as they pass through it on a BWCA paddling trip. And second, they don't have to monitor in a specific location. It's just any lake they happen to be paddling through when the conditions are right. Getting a water quality clarity reading in the BWCA is pretty tricky because from a canoe, most people don't have an anchor or any way to stay in one spot while taking the Secchi disc reading, and it can be really difficult in high winds to get a decent reading. Next slide. Lake ice reporting. So this is one of the science opportunities that the MPCA provides. It happens to be a collaboration with the Minnesota State Climatology Office. The Climatology Office has been collecting ice data on lakes across Minnesota since the 70s, but until recently, they focused mainly on ice off on a select group of lakes. Back in 2003, a research scientist at the MPCA saw a presentation from a limnologist on the effects climate change was having on lakes and asked the limnologist what was one of the most important pieces of data we could be collecting to track climate change's effects on the lakes. And the limnologist said that ice on, ice off dates was most critical. So then the MPCA did start collecting this data, and rather than replicating the efforts of the State Climatology Office, they just shared their data directly with them and making sure that it was a joint effort instead of competing programs. The MPCA's participation in lake ice data collection has increased the number of lakes across the state, with ice data being collected on them and feeding the Climatology Office's repository of data for their own analyses and other scientists to use too. Next slide. So those are the citizen water monitoring programs that are available with the MPCA. And now you might be wondering, who can participate? Well, basically anyone can become a citizen lake or stream monitor or ice reporter. We find the programs are particularly appealing to retirees and families that live on or near lakes or streams. Next slide. You may also wonder why people sign up to participate in the program, and the top reasons given are usually curiosity and concern for their favorite lake or stream, but oftentimes a particular water body has played an important role in the life of them or their family, and they just want to contribute to a better understanding and or protecting it. Other times people are genuinely interested in just the science-based process and maintaining their connection to contributions through data. Next slide. So what it really comes down to is that our citizen water monitors are seen as important water advocates in their communities. They possess valuable place-based knowledge. They oftentimes have emotionally or financially invested in the water body because they often live right next to it or it's a feature in their community. And they are the first to feel the impacts of a degraded water quality on their favorite water bodies. Next slide. Not only are the water monitors local advocates, but they are also important data collectors. Their data is directly used in the MPCA's water quality assessment process, so it's not just data being collected to sit in a repository in the hopes that someone uses it. The MPCA actually does use it regularly. And citizen water monitors collect more than just usable data. They extend the MPCA's capacity far beyond what staff can actually accomplish on their own, and that also includes SWCDs and local partners. So with over 12,000 lakes and 92,000 miles of streams in the state of Minnesota, staff simply just can't monitor all that water on an annual basis, and that's where the citizen water monitors come in. They are invaluable every year in collecting critical data to prevent data gaps and also contribute to the longevity and be able to assess trends. Next slide. So this one's a little bit more on a visual of citizen water monitors and how they provide a return on investment. Just in 2019 alone, 939 lake monitors contributed 4,820 service hours and contributed to an economic impact of $122,573 for the state of Minnesota. For streams, 423 volunteers contributed 3,060 service hours, resulting in an economic impact of $77,816. And that's pretty astonishing, especially since the state provides the kits, which are only about $50 per kit. So it's a really good return on investment for the state. People contribute so much more than they even realize. Okay, next. So the data that they collect, governmental agencies use this data to better understand and protect and restore lakes and streams in Minnesota. The volunteers mainly collect water clarity data, although it's a simple, easy test. For lakes, so water clarity can provide an estimate of algal levels. It can show a lake's response to seasonal changes in temperature and precipitation. It can show a lake's response to surrounding land use changes, and it can also show a lake's response to one-time events, such as a severe weather event, construction, or new developments. Next slide. This SECI data, combined with phosphorus levels and chlorophyll A levels, also known as the amount of algae in a water sample, can help classify lakes into one of 4 different main components or categories. Oligotrophic lakes are the super clear, nutrient poor, deep lakes generally. And then it goes on a scale up to hypereutrophic, which is extremely nutrient rich, shallow, and very green with an overgrowth of algae. Next slide. Stream water clarity data also tells us about the amount of light penetrating into the water body. So that kind of indicates what kind of plant life will be found in the stream, the composition of the stream bottom and the sediment, and if that is the limiting ability for plants and plants and insects to thrive. Next slide. So you may be wondering what causes low water clarity. For lakes, it's mainly an issue of overgrowth of algae caused by an abundance of phosphorus in the water, which can come from many sources, including atmospheric deposition, wastewater from industry, fertilizer applications at home, manure runoff from farms, and occasionally sediment can be an issue for lakes, but its clarity is mainly an algae issue for lakes. Sediment is more of a contributing factor for decreased water quality in streams. The main sources include stream bank erosion and surface water runoff from the local surrounding areas. Now that we know the kind of data citizens collect and what it means for the health of lakes and streams, here's how it's used. Citizen collected data directly informs watershed management at the MPCA in two main ways. First, by contributing to the large data set needed to detect water quality trends over time. And second, by contributing to the data set needed to determine if a lake or street meets impairment standards. All right, so this is where it gets a little data heavy here, or just a peek glimpse inside of the data.
And Kim, I'm just going to interrupt very quickly that we are a little short on time here. We got 3 minutes left, so we are going to skip that data portion, I think, here.
Right on.
And
okay.
We'll go on.
Get to, yeah, it's a lot.
I want to have time for Rachel, so if we could.
How about slide 21?
I'm not able to see the slide numbers, I'm sorry.
Yeah, sorry. Okay. Well, they're all good slides, even if they're just.
Sure, and is this still? So I think if it's okay with you, Kim, maybe we'll move on to Rachel. I know some people probably do need to leave, and it is almost 1.
Sure.
So I'd like to move on to Rachel if I could, and just say that Lindsay has put the link for the ability to enroll as a citizen stream monitor, and we'll have that link as well on the website along with the this recording. Sorry to cut you off, Kim. Thank you so much.
That's okay. I knew it was going to be a long one, so.
No worries. Okay, so I apologize, Rachel. We're running short on time, but I'm going to hand it over to you. Thank you.
Yeah, no problem. Sorry about that. We'll try and keep this really short. So I'm Rachel Lindgren. I'm a hydrologist with the Minnesota Department of Natural Resources, and I'm going to be just kind of briefly sharing with you all a little bit about the Minnesota Spring Inventory and our citizen application. Go ahead and go to the next slide. So what is the Minnesota Spring Inventory? It is a database of spring locations across the state of Minnesota. So me as a DNR staffer, we kind of had to narrow down our focus on what areas we could feasibly get to. As you guys can see, state of Minnesota is pretty big. There's a lot of area to cover. So as DNR, we decided to focus primarily on publicly accessible land. So state parks, trails, water management areas, wildlife refuges, places like that. So areas that are accessible to the public. So that leaves a huge portion of the state that we don't get to cover. And that is where the public can be involved with our citizen science application and using our spring inventory application online. Just a quick thing, when you go to our website, mndnr.gov/mn spring inventory, the map that is pulled up with our webpage, all of the blue dots that you see on the map are spring locations that are currently in our database. They are interactive, so you can click on them and see maybe the date it was visited, if there's any photos associated with it, and other information. So currently in our database, there are 5,160 springs, but there's definitely more to be found. So we can go to the next slide. So why are springs important? They are a major part of our water resource system. They can kind of show a connection between surface water and groundwater. They can show a good window into the health of our groundwater system. And Minnesotans are known for their fishing. Springs are an essential supply of cold water for Minnesota trout streams, and they provide unique ecosystems for things like calcareous fens in the state of Minnesota. So we can go ahead and go to the next slide. So the online reporting application, it is accessible on our webpage. Like I said, Minnesota Spring Inventory. It can be accessed on any type of mobile device, phone, tablet, or also desktop computer. It can be run on Android, iOS, Windows systems. And if you don't feel like using that online application, you're more than welcome to email the Minnesota Spring Inventory itself to let us know of any locations that you found. So I'm just going to do a quick overview of what the reporting application looks like. As you can see on kind of the right-hand side of the spring, when you click on the reporting app, this is what will show up. And there's just a few things that we ask because they can kind of help lead us to determining if the spring location can be added into our database. So we ask for the date that you may have seen the spring itself. That could have been yesterday, that could be today, and that could have been 30 years ago as well. We say, you know, if you remember swimming in a stream as a kid and you know that there was a spring nearby, we welcome those locations as well. Sometimes springs have common names like Big Spring, Fountain Spring, something like that. So if it's known to the public by a common name, you can write that into the spring name field. And neither of these fields are required, so you can still submit this without filling out that information. We can go to the next slide. Springs are known to be flowing, can sometimes flow kind of peak seasons, so they might not be flowing all year round. So we just ask that you let us know if the time that you were there and saw the spring, if it was flowing. It's just a simple yes or no question. And then we kind of classify springs as different types of springs in our database. And we have examples of different types of springs with some pictures on the application just to give you a guide of what spring you might be looking at. And if you don't know, you can feel free to leave that blank as well. Some really common ones are stream bank springs or lake springs. So those are probably the most common that get submitted. And then there's a general comments field as well. And that just has some, if there's anything that you want to let us know, name, email address, and phone number are not required, but they let us know who you are, and we like to send a quick thank you email to people as well. If you have a phone or tablet, you can attach a picture of the spring as well. We can go to the next slide. And then selecting a location. This is by far the most important thing when submitting this to our spring inventory. If you have a phone or a tablet out with you while you're hiking or walking or out on the lake, you can just hit the locate me button and that will just ping your phone's GPS. If not, if you're at a desktop computer, you can type in the address and it will ping to that location. I think we can go to the next slide. That's pretty much everything. Sorry, that was so rushed. I would like to say in the 2 years that we've had it up and running, we've already had 350 submissions by the public into our database. So makes up a good percentage of what we have right now.
Rachel, thank you so much. I thank you for going so quickly, and you did a great job. And Kim, I'm so sorry to have cut you off. Thank you for your patience there. To wrap up Kim's, I want to mention again, Lindsay did post the link on to the website for more information. It was recorded, so there is a recording if you had to leave and you missed something. I would like to, it looks like we still have quite a few folks here. If we have time, presenters, we could take a question or two, even though we are over just a little bit. Would that be all right?
This is Dick Locker. I have a comment.
Okay, go ahead.
An interesting thing about the Big Fork River, as long as we're talking about it, there was some drought conditions here a few years back when we noticed that the bog stain in the river had completely disappeared. So it turns out a substantial portion of the volume of water in the Big Fork River is in fact from springs.
Yeah, that really doesn't surprise me. A lot of Minnesota is covered in kind of large wetland bog conditions, and those are what we would consider like groundwater seeps, so kind of large spring complexes that feed those ecosystems.
Yeah, that's wonderful to know, and hopefully we can find some of those springs and get them sent over to Rachel's way so we can get them on the Minnesota inventory. It would be great.
Excellent. All right, it looks like we just had 2 quick questions in the chat, and I'll go through those quickly just so that those that weren't here can hear answers for them. David Lick asks, what are the state agencies doing as far as a policy with trying to change the continuing impairments? It seems like we ID the impaired waters but don't require stricter guidelines. That might have been a question maybe for Jesse or Megan, I'm not sure.
Lindsay, I don't know if you have more of an answer. Because unfortunately, yeah, we just do kind of the condition monitoring, like you said, identifying them, and that's kind of where that goes. The next phases are kind of where Lindsay comes in, and I know there's the buffer law that's been put in. That's kind of a way. Unfortunately, I guess I'm not sure how much this is situation for the Big Fork, but unfortunately, in a lot of parts of Minnesota, we know that agriculture is kind of a big impact, and there's not a lot of regulation that goes there. I guess that's what I got for that.
Yeah. And I can add too just a little bit. And so following up after we do the assessments and then the lake or stream gets put into that impairment status, we do follow up with some stressor ID to figure out some further investigation to try and figure out the causes. And then all of this kind of gets wrapped up into our protection and restoration strategy report. And then that is taken further by stakeholders and local units of government that can start putting them into maybe their county water plans and try to get some funding for implementation activities that can be done on the ground to try and resolve that impairment. I would say that probably is the best thing that we're doing right now to try and take care of those impairments. Angeline, you're on mute.
Thank you. So I was just saying thank you, Lindsay and Megan, and thank you for that heads up. I just have one more question in the chat that I'm seeing here, and it was Tom Nelson. Are the lake level variations over the summer via a gauge zeroed in by the DNR staff also a part of MPCA citizen monitoring? And because we don't have Shannon Martin here on the MPCA citizen monitoring, I'm not sure if we know the answer to that or not, or if we could maybe get back to him with that.
Yep. Yeah, that's exactly right. So Shannon's not here to answer those more specific questions, but I can follow up with her and then send out an email and address those questions. So
This is Jim. Address some of that. We asked the citizen stream monitoring folks to give an estimate of the river level. Some folks actually measure it with a tape from a fixed mark on a bridge. Other folks just put it into 3 or 4 categories based on their opinion. Is it high, normal, low? It's pretty rare for a citizen monitor to be monitoring at the exact same location of where the DN has lake levels or stream levels being monitored, but we do try to connect if we know that.
This is Dick Locker again. I also do lake level monitoring, and I monitor in Dora Lake, which is essentially where the Poplar River and the Bowstring come in and the Big Fork leaves. So it's a good indication of water level for the river at that point. And that's from a bridge abutment.
Thank you. Thank you, Dick, and thank you for the work that you do with the students. I know that's a long-lived program that you've been running, and it's very helpful. All right. That is all I have for questions here. And I think what I will do is if there are further questions, you can certainly connect with us at Kuching SWCD, and I can reroute the question to the appropriate person. This has been recorded and it will be located by tomorrow, likely sometime tomorrow morning, at the same website here that you registered at. And instead of the register link, it'll say watch this video. We're also going to post those links and the information that we've been talking about here and Lindsay has been posting in the chat on the website so that you have it as resources as well. So I want to thank you all for staying longer as well than we planned, and thank you for coming and spending your lunch break with us on the Big Fork River Talk.
Thank you.
Thank you. Have a
Thank
great day.
Thank you.
Thank you.